So we were talking about the nucleus last time, and I already explained what exactly you have to know. Everything that I'm focusing on is important. So what do we need to know about the nucleus? I told you the nuclei inside the nucleus, nucleolus inside the nucleus. What is the importance? This is where we make RNA, which is part of the ribosomal subunits. Very important. This is the function. Nucleolus is responsible for this, making RNA and putting together the ribosomal subunits. Um, Next is nucleosome. Nucleo is nucleus, some is the bodies. So what are the bodies inside the nucleus? There is two types, and both of them are basically the same. The only difference is how they are coiled, the way they were coiled. Both of them are, which is chromatin and chromosome. Both of them are DNA wrapped around histone, a protein. But is it loosely tied or tightly? Is it loose or tight? Okay, so this is something that's important to know. Chromatin is loose and chromosomes is tight. And chromatin is loose. This is if the cells are not dividing. It's not the time to make it tight and start the reproduction or the division, I mean. If it is time for division, so the vast majority of time, this is non-dividing. The cells are not dividing all the time. There is a big time where the cells are not dividing. If it is the time, when it is the time for dividing, this wrapping will become tighter, to become more tight, and now we call it chromosomes during cell division only. So if you look at this picture right here, this is the cell, this is the nucleus, and you will see the chromosomes inside. This is one of the chromosomes. And what's a chromosome? Again, DNA wrapped around protein called histone. But the chromosomes consist of two sister chromatids. So what's a chromatid? Half a chromosome. And look how they are super coiled around these histones. Look at the histones. Okay, the histones are the blue ones. And what's wrapped around it is DNA, which is a double helix that we talked about. But if you get a bunch of those histones with the DNA wrapped around it, we call that nucleosomes. So you got a couple of definitions that you have to know. Now the genetic code. I mentioned before that the DNA is where we store our genes, our genetic information for inheritance, plus it contains instructions to make all types of proteins. Clear? This is where we make all types of proteins. The instructions are on the DNA. Okay? And we talked about proteins before. We said, what's the building unit of protein? What's the monomer of protein? What's the monomer of protein? Amino acid. So in order to make protein, you need to put group of amino acids beside each other, right? So if at this time I want to make protein number 5, how to make it? You go to protein number 5 instructions on the DNA, and you take a copy of these instructions only for this specific protein. What is this copy called? Huh? No, if I wanted to make protein, anything, protein, which type of RNA? Yes, messenger RNA or mRNA. So here is the DNA. This can make protein 1, 2, 3, 4, 5, whatever, 20. Okay? So I want to make protein number 5 because we need it now. What should I do? Of course, I'm not getting the whole DNA. I'm not getting a copy of everything. That's not right. What we will do is we will look for it. Where is 5? Here is 5. So these are the instructions for protein number 5. I will leave everything else alone, and I will take a copy of these instructions only. This copy that I got is called messenger RNA. Small enough to leave the nucleus through the tiny pores. DNA doesn't leave. But the point here is, within the messenger RNA, what exactly tells us what kind of amino acids? Remember, protein is a group of amino acids, isn't it? It's amino acids. So which amino acids? Like I want to make this protein, number 5, number 4. I don't want to say names. Whatever the protein is, I want to make this one. Well, you have to put these amino acids specifically in that sequence. And who is telling me that? The instructions. How? This is the genetic code. So the four bases that I talked about, remember the ATCG, and I say it in that sequence because A goes with the T and C goes with the G. So there will be something called triplet code. Every 3 of those 4 in a specific arrangement is translated into amino acids. Are we following so far? So let me say instructions for number 5 are like this: ATCGGGCTAGCG, something like this. It goes 3, 3, 3, 3, 3, 3, 3. Every 3 means amino acids. Okay, so this is called the triplet code. Triplet code. Every 3 bases, ACG, it means specific amino acid, and you don't need to know what code and what amino acid. This is too much. Just know the idea of every 3 amino acids. So 3, 3, 3, 3, 3, 3 amino acid, amino acid, amino acid, amino acid, and this is how we put the instructions. Triplet code. Every 3 bases means 1 amino acid. What is the gene then? The gene is instructions to make 1 whole protein. Remember I told you the DNA is a long strand, right? And it codes for all proteins, right? So this is coding for protein number 1, gene 1. This is coding from protein 2, gene 2. This is coding for protein 3, 4, 5, and so on. So if I get a copy of this, I'm getting a copy of a gene. What's a gene by definition? DNA instructions for 1 protein only, 1 protein with a long chain of amino acids. It's a polypeptide. And this is a functional unit of heredity. Now, how can we make protein this way? Protein synthesis. The first thing is, which protein do you want to make? X protein. Okay, I will look for it on a DNA. Okay, these instructions are for protein X. What do you call it? A gene. It's instructions for 1 protein. So what are we going to do now? I'm going to activate this part only, or this gene specifically, in order to be ready to get a copy of it. So we do activation first. How? Remember the DNA is double-stranded, isn't it? And twisted like this. Untwist. You wanted to get a copy of 1 of those strands, but they are like this. So you cannot get a copy, right? If you have a group of papers like this, and you want to get a copy of 1 of those, you have to take them apart, right? So you unfold it, untwist it. So now you have this strand and this strand. And then I will look at that strand and make a copy of part of it, which is the gene. And we call making that copy, and what's the copy again? What do you call the copy when you take it? mRNA, messenger RNA. This is the copy. You call this process transcription. So if I ask you what's transcription, the formation of messenger RNA, which is a copy of 1 protein or a copy of 1 gene. This is the process of transcription. So the DNA is 2 strands? Yes. One of them is called coding strand, and the other one is called template strand. The coding strand is the one that has the code. The template is the other one. And the template is the one that we use to get a copy. Do we take a copy of the whole DNA? No, we take a copy of 1 protein only, right? Instructions for 1 protein only, which is called the gene, right? So we have 2 strands, coding and template. I take it from the template. I take a copy of the template to make messenger RNA. In this process, the enzyme that's helping this process of making messenger RNA is called RNA polymerase, and you need to remember the name, RNA polymerase. Polymerization is to make a copy, polymerize. Okay, you're making a polymer. So RNA polymerase, look at the first part because there is another one similar. It's called what? RNA polymerase. You're getting a copy, and this copy is RNA. Because later you will see DNA polymerase. What's a DNA polymerase? So the first part of the name is the product. RNA polymerase, the enzyme that helps us to make what? RNA. DNA polymerase, the enzyme that helps us to make DNA. Meaning this is DNA, I want to make a copy of it. Does this have to do with a protein? No. It's just duplication, like in cell division or something. So polymerase, there are different types. Read the first part. And this will be the product. RNA polymerase, enzyme to make RNA. So this enzyme is going to attach to the instructions to make one protein. And there is a point where it should start. There is a code that tells RNA polymerase start here, like protein 1, 2, 3, 4, 5. At 5, if I wanted to get a copy of 5, what should I do? When the enzyme get closer and start to touch, there will be a specific point to tell start, something like this. Start here. It's a start point, and there is end here, and then start here for number 6, end here, start here, end here. For each gene, there is a start and end. Otherwise, RNA polymerase will be going like this. 5, 6, 7, 8, he will be copying anything. If you don't put specific points to tell that enzyme where to start and where to end. So here is the two unfold, untwist. Now I have two, coding and templates. I need the template. So I will get RNA polymerase, and it will look for the one that we need. Here it is. Where exactly should I start? You will see a code telling you to start. Copy, 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 until you see end, stop. Now you have messenger RNA. It's going to detach itself now, and it doesn't leave as it is. This copy doesn't leave as it is. You wanted the copy to be clean and organized. Meaning, if I am taking a copy of instructions how to make cake, for example, and there is a big paper like this, and the instructions are only like 3, 4 lines. Why do you need all of this white? You're going to cut it and just have this part clean instructions. Did you get my idea? So you're going to cut away anything that doesn't coat. Why do you need to get a bulky thing? You want it to be as small as possible so it can easily pass through the pores. And we call this process RNA processing. So before you leave, anything that does not code, which is like the blank paper, the white paper, there's nothing written there, you cut it away and get rid of it. And leave only the ones that have the actual instructions. What do we call it? Exons. So here is a whole page, like 2 lines, and then space, white, 2 lines, space. Why do I need all of this? I will cut off all these whites and just put together the ones that have the instructions, 2 lines, 2 lines, 1 line. That's it here. And I get rid of everything else. What do you call everything else? You get rid of it. It's called introns. Okay? It was in between, introns. And the exons are the one that I need. So I get 1 exon, 1 exon, 1 exon, put them together. This is your messenger RNA after processing. Here it is again. Here are the 2 strands, separate and untwist. Here is RNA polymerase. It will find the sign to start. And this sign is called the promoter or the start point. And then you read 3 by 3. Okay, TAG. It means amino acid. You don't have to know which one. You will see it, but you're not responsible for it. And then you see GCT. Okay, copy. and so on. You'll get copy, copy, copy. You're copying the whole thing until you make this, which is called the codon. The copy is called the codon, which is, uh, I mean, the every three are called codon. And so I have codon, 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 and so on. And then this messenger RNA will leave those two strands, move away, detach itself, and the two strands are going to go back together. You're done. Messenger RNA formed, slice introns, get rid of it, keep the exons, put them together. Now you have a small, tiny messenger RNA, clean, ready to go. This was going to leave the nucleus from the nuclear pores and go to the site where we make proteins. What is that site? Start with R. What is it? Ribosomes. Ribosomes. RNA is part of the ribosomes, but it's called, the factory is called ribosomes. The machine is called ribosomes. Remember ribosomes? Remember the free and fixed, rough ER and rough endoplasmic reticulum, smooth endoplasmic reticulum? That was yesterday. So here it is. After you're done, RNA polymerase will leave, and then you have this copy, which is a messenger RNA. This messenger RNA is going to leave through the pores and go to the ribosomes in order for the ribosomes to read the instructions that are clean and ready to use. The ribosomes are going to read it and translate that into amino acids. Remember that every three means one amino acid, right? ATG, CGG, GCC, whatever. You can put it in different ways. So the messenger RNA is going to go out of the nucleus to the cytoplasm, looking for the ribosome, and go to that ribosome. The ribosome itself is two units, small and large units, just like this. This is the small and this is the large. They are together like this. Once the messenger RNA come, they are going to separate so that the messenger RNA go in between. Okay, this is how they translate. So how exactly are they going to translate? They, what is it? tRNA. They are going to use the other types of RNA. How many types of RNA do we have? Three. mRNA, this is the first one. By the process of transcription, right? This is a copy. This is the instructions. It go to the ribosomes. Ribosomes split, small and large, to allow messenger RNA to go in, and then it's go back like this. And we're going to start reading. So I'm reading right now. Okay, we're reading. The first part will say start here. Okay, start here. Okay, start. It doesn't mean anything. I'm just starting. Okay, next. CGG. It means amino acid, histidine, whatever it is, glycine, any amino acid. So, okay, now we're reading the amino acids. Who is going to bring it? We have it stored. Transfer RNA is going to transfer it. Okay, so we're reading. Let me say I am the ribosome. Okay, I am the ribosome, the two units. Messenger RNA came to me and I start reading. Start here. Okay, next, CGG. CGG, it means glycine, according to my memory. Okay, glycine. Then I'm calling on transfer RNA. Transfer RNA. Which one of you guys is responsible for glycine? You? Okay, bring it. Okay, you will get it. You have all these amino acids sitting there, like on the shelves. So transfer RNA, please transfer the amino acids and bring it here. Then I have to double check. Are you sure this is the one that I need? Yes, it is. No, I can't take it from you. I want you to fit yourself on the code to make sure that what you're bringing is correct. So let's say the code was on the messenger RNA, CGG. Show me your anti-code. Are you bringing the amino acid? Yes. Before you leave, I want to make sure that this is the correct amino acid. How? Just put yourself here and see if you fit with my code or not. Okay, this is like when you go uh, to a store and make a copy of your key, house key, right? Don't you need to like try it first and make sure this is the correct one, it's working, right? So are you bringing the correct amino acid? Yes. I have to double check. Put yourself here, fit, see if you fit or not. If you fit, you're correct. Leave the amino acid and go. Okay? How to fit? Remember the codon is these triplets on the messenger RNA, right? The transfer RNA has an anti-codon, something that fits together like this. Okay? Here is the codon on the messenger RNA. Here is transfer RNA coming and bringing the amino acid. Did you bring the amino acid? Yes. The correct one? Yes. Still double check. Fit yourself in. Yes, it's correct. Leave. Okay? So the codon is the codes on the messenger RNA. The anti-codon is the code on the transfer RNA. They has to fit together to make sure. If you're sure, okay, leave. I got one amino acid. And then I read the next one. What's the next one? GAT or GAU. Okay, GAU, it means histidine. Who's transfer RNA responsible for histidine? Me. Bring it. Come here. Is it the correct one? Yes? No, still fit yourself with your anticodon. Does it fit? Yes. Now I know that this is the correct amino acid. Put it beside the other one and link them. And what do you call the link? Peptide, right? You have a question? What's what? Okay, that's a good question. We talked about messenger RNA, the copy. We talked about transfer RNA, the one that transferred the amino acids to us. rRNA, ribosomal RNA, is the main component of the ribosome itself. Okay, it's the ribosome. The ribosome is ribosomal RNA plus other things, plus proteins. You get the idea? I have three types. So, got the first amino acid, double checking, codon, anticodon, correct, leave. Next one, bring it, amino acid. Now I have two. I'm not going to leave them like this. Make a peptide bond. Okay, now I have monopeptide. Next, bring the amino acid, fit in, yes, link, peptide, 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 peptide. I will end up having a polypeptide. Did you get the idea? This is the idea of protein synthesis. I just I'm just summarizing, but this is more than enough what I'm telling you. Okay, these are the codes, and of course you don't need to know the codes. Okay, just look at it to understand how does it work. So if this is the DNA, it was triple A, adenine, adenine, adenine. This is a template, and the coding strand was TTT. Okay, so the copy will be UUU. How did you know that? Because A. What goes with the A? What T, right? This is for the DNA. Do you think RNA have a T? No. So what fits instead? U. So if you have A in DNA, what's a correspondent one in the RNA? U. Is that clear? If it's A, your copy is U, because RNA doesn't know the T, doesn't have T. So this will be U. Okay, now the transfer RNA is bringing that amino acid, which is phenylalanine in this case. U means phenylalanine. So transfer RNA is bringing phenylalanine. Okay, is that the correct one? Yes. Okay, fit your anticodon first. What's your anticodon? A. It fits with U. Yes? Good. Leave it and go. Go to the next. The next is UUA. It means leucine. Transfer RNA. Transfer amino acid to me. Did you get the leucine? Yes. No. Fit your anticodon first. AAU. Let me see here. A go with the U. A go with the U. U go with the A. Yes, correct. That's correct one. Leave it and go. Okay. You don't need to know those again. But just this is just as an example to see how it looks like. The codes, no, you're not responsible for it. I'm just showing you how it looks like. But what I can ask you is what fits with what. That's the only thing that I can fit you. Like I can give you A, G, 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 C, C, for example, for the DNA, and ask you what's the RNA that fits with it. Okay, so A, G, 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 that will be U, C, 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 right? This is the codon on the messenger RNA. Or I can give you a codon and ask you for the anticodon. Are you following? So it's U, C, C, C. What would be the anticodon? A, G, 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 right? So this is the only thing. What fits with what? That's it. But the codes itself, too much. Okay, so if you look at this blue here is a ribosome, the blue, and it consists of what? Ribosomal RNA. So here, look at this. Here is the messenger RNA coming, and the first thing is AUG. AUG means start. So this is not going to translate. And then I will read the next. Look at the transfer RNA bringing the amino acid. Can you see this? Transfer RNA transferring the amino acid, transferring the amino acid. Fit yourself. Codon, anticodon. Yes, leave it. And then next, number 2. Fit yourself, leave it. Okay? And number 3. But look what happened between 1 and 2. What is this? What did I do here? Peptide bond. Yes. And the third one will come, link it, peptide bond. Okay, so here is, what do you call this from 1 to 7? What do you call this product? Polypeptide. Yes, it's called polypeptide. Okay, now we're talking about protein synthesis, and we know the instructions on the DNA that are called genes. Each gene is a code for what? The gene is a code for what? If you're following with me. For one protein, yes. One code, one gene is a code for one protein. Is that clear? The triplet code is for one amino acid. Clear? Okay. So now this is DNA. I have all instructions for all proteins. Remember I told you that we will get a copy of this specific protein? Who decided that we need this protein right now? Who decided? Instructions coming to the DNA. Who is telling the DNA? Unfold, untwist, expose, make a copy, and all that. Who give the orders? DNA is the one that gives the order, but who told the DNA what do we need? It is a messenger that came to it. There is a messenger come, like the cell needs collagen, okay? The cell need collagen. Are we following? The cell need collagen. So a messenger will go to the DNA. The cell need collagen. Okay, this is protein number 12. Okay, I'm gonna unfold, make a copy, send it to the ribosomes. So messengers go to the DNA. It can come from another cell if needed, right? Do you remember we said we make proteins, some of it for using inside the cells and others for export? So if there is another cell or tissue or organ that need a specific protein, it's going to send a messenger, and this messenger is usually called a hormone that goes, cross the wall, go to the nucleus, go to the DNA, tells the DNA what do we need. Hormone is one of the messengers, the most common messenger. It's called hormone. And we will talk about this later. So this is for the protein synthesis. The next part is about the cell membrane that we talked about before. Remember that 2 layers of phospholipid? So the cell membrane can allow certain things to go in and out, and others are not allowed. Like this room, not everything, not every class is allowed to come in here, right? There is a door. I have to use this to open the door for me because this is my allocated time, right? So I can come in. Anybody else? No, you're not allowed to come in. So this is called selective permeability. What's permeability? Permeable is permission able. You got a permission. So is this freely permeable? Anybody can go in and out? No. Is it impermeable? I am at the beginning means no. Nobody can get in. Is that the case? How did you get in? No, it's none of those. It's not freely permeable and it is not impermeable. I am, I-N, A-N at the beginning of any word, it means no. Okay? No. So impermeable, no permission. Is that the case? Of course not. So is it freely permeable or impermeable? None of those. It's none of those. It's not free and it is not impermeable. It is selective permeable. Selective, in between. Certain things are allowed, others are not. So our cell membrane is selectively permeable. It allows certain things. Here it is. It allows certain things to go and others will not go. How the membrane know that this is selected to go? How did you know? How the membrane know that this is okay to go and this is not okay to go? Like I am okay to go because I have this, I can just scan it and go, right? How does the membrane know which one is allowed and which one is not? It will depend on whatever is coming and trying to cross. What's your size? If your size is small enough, yes. Electrical charge, the shape, molecular shape, and lipid solubility. These things, four things together, which is important to know, determine who is selected to go, depend on these variables. Now, if you wanted something to cross the membrane, if you are okay to go, you have a permission to go, okay? And here it is, it's coming to the cells, and I want to cross the membrane, right? What are the different ways to cross the membrane? It's not all the same. Some of these transports does not use energy. Okay, it's you just cross the membrane and go. You don't need energy. Others need energy. The ones that does not need energy is called passive. The ones that need energy are called active. And you do need to know these details, including which is passive and which is active. Okay, we will talk about the different types, and each one of those you need to know if it is passive or active. How to know it's passive or active? Passive does not use energy. You just pass. We call it diffusion. Just move. No energy needed. Others, you have to have energy. So diffusion and osmosis, those two are passive. Okay? And I will tell you what I mean by this. The first two ways of crossing the membrane are diffusion and osmosis, and this is passive, meaning no need for energy. There is one that's called carrier mediated. This can be passive or active, depending on variables that we will talk about. The last one is definitely active. You have to have energy to do it. And this is called vesicular transport. Do you guys remember the vesicles that we talked about, secretory vesicles? Like if this is for export, you go to the membrane, you fuse with the membrane and go out and then reform the membrane. That was, I think, yesterday. Okay? In order to do that, this is too much. And let me say it this way. If I want something to come in here, there are different ways, right? If I'm coming by myself, I just scan this and go, right? If it is something a little bit bigger than that, you can barely go. What if I wanted to bring a truck in here? I can't get a truck. What if I have to? I need it in the class. What should I do now? I'm going to break down the wall. Allow the truck to come in and then reform the wall again. Of course, that needs energy, isn't it? This is the vesicular transport. So if there is something that does not, if there is something that does not that just goes with crossing the membrane and doesn't destroy or doesn't do anything. Just move in and that's it. You didn't do any changes. This is something that's passive. If you are going to do changes like you're going to break down the wall, in this case you're breaking down the cell membrane. Literally, it's too big that you have to break the cell membrane, put them in a vesicles, move in, and then reform the membrane. Break the wall, trucks move in, rebuild the wall. That's exactly what it is, and we call that vesicular transport. Diffusion, simply, key, one word, concentration gradient. If you move something from high to low, it's diffusion, it's passive. That's it. Okay? Diffusion. What's diffusion means? You're moving across or via concentration gradient. If this is high and this is low, it will just diffuse from high to low. I have a lot of sodium outside of the cells and a little bit of sodium inside. Can the sodium move in? Easily. They just diffuse in. Did you get the idea? It is passive because you go from high to low. So what kind of effort do you need? Why do you need energy? You're just diffusing in. This is high and this is low. That's okay, right? Like if you use your bike and the street is not straight, it's going like this down. Do you need to make any effort? You would just move in, right? This is exactly what it is. You go from high to low. It just diffuse in, right? Here I have 100, here I have 20. From 100 to 20, it's moving in. Yes, diffusion. Okay? So concentration gradient. And it is something like this. If I put this colored material here, it's going to move from high to low. So it's going to diffuse. This is if this is a solution. If this is the membrane, it's the same idea. Crossing the membrane, using concentration gradient or with concentration gradient, this is called diffusion and it is passive. You need to know both of them. Diffusion, concentration gradient. You're using concentration gradient from high to low. Clear? If that's the case, do I need energy? No. You're moving from high to low. You don't need energy. Diffusion. Okay? What determines how fast the diffusion occur? Like this is crossing by diffusion. And another thing is crossing by diffusion. And another thing. They do not cross the same speed. What makes the difference? Size is one. The distance that you're crossing is one. The difference, the gradient. Are you talking about 2010 or 110? Is it the same if you're driving your bike and the sloping is just like this? Is it the same as if the sloping like this? Of course not, right? If you're going like very high to low, you go very fast, still without doing effort, without energy, right? But if it is going like this, you still don't use energy, but you go slow, right? What if your size is tiny versus if your size is large? It's also different. If me and you are driving bikes on the same slope, right? And my body is bigger than you will go faster. Make sense? Right? It's your if your body is less or smaller, you go faster. So size, distance, concentration gradient, electrical forces is another thing. Are you crossing something that have electrical gradient that help the movement, like this is positive and inside is negative? Yes, that will help as well. Also, if you put some heat, like this is adding, that will make it also faster. So these are the different factors that make the diffusion faster. It's um, affect the diffusion. So diffusion, where exactly do you diffuse? What do you mean by diffusion? Like crossing the membrane? Yes, we know the structure of the membrane. Where exactly do you cross? Do you cross like in between the two layers of phospholipid? Like here is one and here is one. You cross in between, or where, or from the protein. Do you remember the structure of the plasma proteins? We talked about it. So which part exactly are you crossing, from here or from the door or from here? Where? So if you are crossing just from the two layers, just crossing in between, you don't need anything special. This is called simple diffusion. Simple diffusion. You can cross just in that regular membrane, the two layers of phospholipids. And what can do that? Anything that's lipid soluble, or gases that are also lipid soluble. They can do the simple diffusion. Do you guys remember what's the main structure of the membrane? Two layers of what? What kind of lipid? Phospholipid. It's phospholipid. Two layers of phospholipid. Phospho what? Lipid. Lipid. So in order to pass easily, you have to be lipid soluble. Okay? The membrane is lipid. To cross it, you have to be lipid soluble. So lipid soluble compounds, gases can cross like that. Water can also cross, but not the same way. There are channels for it. And we still call that simple diffusion. The other thing is channel mediated. So can I go like others, including ions and water? Can I just cross, just like what you talked about before, lipid soluble? No, you cannot. If you try to go and cross the two layers of phospholipids, you cannot. You're not lipid soluble, but we need it inside. How can I go? Yes, there are channels for you. If I need you, you go through channels. So does it need energy? No, still doesn't need energy, but the only difference is you, lipid soluble, you can just cross. You are not lipid soluble, you cannot, but it's okay. There are channels for you. Just move in the channels. And we call that channel mediated, like water and ions. So you do need to know what does it mean and what are the examples for this part. Okay, so here it is. If something lipid, look at these. Anything that's lipid soluble will go like this. Just cross. You see these two layers of phospholipid? Just cross. This is simple diffusion. Crossing those. Um, what? How about water? Is water lipid soluble? Water, lipid soluble? No, it is not. So can you cross? You cannot cross the lipid. You have channels. So you go through channels. Same thing for the ions. Next is also passive, which is called osmosis. Osmosis is something unique for water. When you say osmosis, you're talking about water. That's it. Nothing else. Osmosis is water. And simply, water move from high osmotic pressure to low osmotic pressure. What do you mean by osmotic pressure? Here it is. If I have this container and there's water here, right? And we call the water what? We call it solvent or solute or solution. Water. Solvent. You're adding solute, some salt. Isn't it different when you put 1 spoon versus 5 spoons? If you put 5 spoons, it will be more concentrated, right? We call that higher osmotic pressure. We don't say more concentrated. We say higher osmotic pressure, meaning more solutes. Are we following? Okay. So here is a bigger container, and I put a membrane right here. Okay. I got one container. I split it with a membrane. This membrane is permeable to water. Water can move from one to one. This is number one, this is number two. Is it going to move from one to two or from two to one? I don't know yet. What's the difference between those two? I'm telling you the difference now. Here is 1 liter of water, here is 1 liter of water. Same amount of water. Still, I don't know, right? If you have 5 spoons here of salt and one here, the water will move like this. So it moves from low osmotic pressure. I'm not talking about the amount of water. From low to high. From less concentrated solutions to higher concentrated solution. Did we get the idea? Right?
So it's gonna move from 1 to 5.
We call this part here, we call it number 2, and this is called 1. It will move from container 2 to container 1 because this contains 1 spoon of salt and this contains 5 spoons. Let's make it more practical examples. I get this juice that's diluted, right? There is some juice that's cheap, it's very diluted, right? And there is juice that's concentrated, right? So I get this container, put a membrane, and put here the cheap juice, and here the expensive juice. The expensive is more concentrated, right? Where the water going to move? From the cheap to the expensive, from the diluted to the more concentrated. Did you get the idea? How about juice and honey? If there is juice here and honey here, it will move from the juice to the honey because the honey contain more solute. Did you get the idea? And so on. If this is sugar in water and this is sugar in water, this is very sweet, contain a lot of sugar. It will go from this to this, from low to high. Okay, so we call this osmosis, and this is important for a reason. Here is the osmotic pressure that I was talking about. What's the osmotic pressure? The pressure that forms by adding solute to the water. The more solute you add, the higher osmotic pressure. Is this point clear? The more solute, the higher osmotic pressure. So remember this type of pressure, osmotic pressure. This is one type of pressure, osmotic pressure. There is another type, which is the pressure of the water itself. We call that hydrostatic pressure. Okay? Like if you want water in a balloon, the balloon is going to expand, right? Add more water. This is water pressure, right? Expand. Add more, it's going to burst. Why? Because I'm increasing the pressure of the water itself, the physical pressure. So there are two different types of pressures, osmotic and hydrostatic pressure. The water channels, just for your information, the water channels that we talked about are called aquaporine. Aquaporins. Aqua means water. Porins, pores. The pores for the water, which is referring to the channels. So, osmotic pressure is also known as osmolarity. Osmolarity. Put more solute, higher osmotic pressure, higher osmolarity. Less solute, low osmotic pressure, low osmolarity, which is the osmotic concentration. Okay? So now this will bring us to an important point, which is tonicity. It's basically the same. Here is what I mean. Here is one container, water. Here is one container, water. Gallon, gallon. Same thing, okay? If I put 5 grams here of sugar and 5 grams here of sugar, these are exactly equal, isn't it? The tone is equal. We call that isotonic. Iso means same. Isotonic. Clear? The tone here is the same as tone here. This is gallon, this is gallon. This is 5 grams, this is 5 grams. If it is exactly the same, we call those two isotonic to each other. Clear? How about if I put 1 gram here and 5 grams in this one? Both of them liter, liter. But 1 gram versus 5 gram. Is it the same? There is one that's less than the other. The one that have 1 gram is less, isn't it? We call it hypotonic if you compare it to the other one. Is that clear? And we call the 5, the one contain the 5, hypertonic if compared to the one that contain 1. Is that clear? Did you get the 3 terms? Iso, hyper, hypotonic. It all depends on the amount of solute, which we call it the osmotic pressure. So, what will happen if I put a container like this? Here is a container, all right? And this container contain water and salt. Put some salt in it. Okay? And then I get one of the red blood cells, take one red blood cell and put it in here. The inside of the red blood cells, it also contains water. Do you remember in the very, very beginning? Water is everywhere. Inside the cells, outside the cells, in the blood, everywhere, right? So what's inside the red blood cells? Water. Is it pure water? No, water with other things. We call it watery, watery fluid, not water. It's watery. So water and other things, right? So now we have osmotic pressure in the red blood cells and osmotic pressure outside of the red blood cells. What if the osmotic pressure inside and outside is the same? What do you call it? Isotonic. So if I do this and I put this red blood cell inside a solution that's the same osmotic pressure or the same osmolarity, we call it, you put it in isotonic solution. What's isotonic solution? Solution that have the same tone, isotonic, of the red blood cells. Is that clear? What's going to happen? Water moves from the solution to the red blood cells or from the red blood cells to the solution? What do you think? Nothing. Yes. It's not moving. Why would you move? This and this are exactly the same. Why? Remember the osmosis? What's the osmosis? You move from low to high concentration. Where is the low and where is the high? Nothing. Right? It's the same. So what happened to this red blood cells? Nothing. Okay? How about if I get this same red blood cell and I put it in this solution and this solution is concentrated, higher concentration. So you put it in what solution? What do you call the solution? If it contain more solution. Hypo or hypertonic? Hypertonic. So what happened if I put the red blood cells in hypertonic solution? The water moves out of the red blood cells or into the red blood cells? Out. Yes, out. And the red blood cells are going to shrink. We call that crenation. And we do need to know this word, crenation, shrinking. How about the opposite? What if I put it in hypotonic? What do you think will happen? How the water moves? But how does it move? Into the cells or out of the cells? Into the cells. So what's going to happen to the cells? Swell, 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 rupture, hemolysis. We call that hemolysis. Did you get those two terms? Crenation and hemolysis? The third one is nothing. Nothing is going to happen if it is equal. So here it is. Nothing happened versus in hypotonic. It's going to swell, then rupture, hemolysis. In hypertonic, it's going to shrink, and we call it crenation. This is for the first type, which is passive, which is diffusion and osmosis. Clear? Diffusion and osmosis. This is passive. Done. Next one is carrier mediated. Is this carrier mediated? Is that passive or active? It depends. This one specifically, it can be active or passive. Diffusion and osmosis is passive. No exceptions. Okay? Vesicular transport, active. No exceptions. This one is in the middle. It can be. So this one is simply something that needs a carrier to take it across. It cannot go the regular ways. There should be a carrier to carry it from outside like this to the inside, or from the inside to the outside. If you don't have that carrier, no way. Nothing is going to move. So we call that carrier mediated, which is basically a protein. And this protein have specificity, saturation, regulation, just like the enzyme. Do you guys remember the enzyme when we said the same thing? So it does have its own way of working. It is regulated. You can tell them move something or don't move something. So this is using a carrier. Okay. Now if this carrier, are we following? We're talking about a carrier that's taking something from out to in or from in to out. Clear? This carrier, if this carrier is moving two things at the same time, not just one, like we need to move sodium and glucose. We need to move both of them, and you're responsible for both of them. This carrier is responsible for both of them. What do we call that? If both of them are coming together, like the carrier take both of them in, we call that um, contransport or co-transport. Co. What's co? Together. Both of them go together on the same way. The opposite is counter transport or antiport. So there is same port, which is co-transport, and there is counter. What does it mean? Here's what I mean. I am a carrier for sodium and potassium. You know what I'm going to do? I will take sodium from outside to inside and potassium the opposite. Isn't that counter, right? I'm moving one in and one out at the same time. This is counter, right? Unlike sodium and glucose, I take both of them, I move both of them in. This is co. Co means together. So if I take, here is the example, glucose and sodium, both of them, I'm a carrier taking both of them from outside to inside, both of them same way, co-transport or same transport or same port, I mean. If I'm doing the opposite, I'm going to carry sodium and potassium not the same way. I'm going to exchange them. We call that counter transport or antiport. So this is called facilitated diffusion. What's facilitated diffusion? It's a diffusion, but need the carrier to facilitate this diffusion. Okay? Something like this. This is a carrier, and we mentioned that the carrier specificity, and saturation and regulation. So this is the carrier for glucose. If you leave the glucose alone, it will never pass. There is no channel for it. There is no diffusion. It's big. It cannot just pass in between. It's not lipid soluble. There is no way that you get the glucose in. The only way is carrier to carry it in. And specificity means this carrier is for glucose. Glucose goes, fit in at the receptor site, and look at this. It will open and move it in. So the one that we talked about is the passive one, facilitated diffusion. The active one is called active transport. In this one, we're using energy. The carrier use energy in this one. And the most important one is sodium potassium exchange pump. It goes like this. If this is the cell right like this, here's your cell, okay? Here is sodium, and here is potassium. Do you mean that there is only sodium outside and potassium? No, I didn't mean that. I'm just giving you an example. There is sodium and potassium outside, sodium and potassium inside. Not the same amount, but you have both of them. Is that clear? So here is transport. This is a transport. I'm going to take sodium out, potassium out, in exchange of sodium in. Okay? This is counter transport, isn't it? In order to do that, there are two things that you need to know. Number one, you have to use energy, or you wouldn't be able to do it. In order to do that, we found that you have to use energy. So this is active. Number two, they found something interesting information that it's not only active, but there is a ratio. Potassium to sodium is 3 to 1. Here is what I mean. This is the cell. Okay? I'm a carrier here. This is the cell with the membrane. I am a carrier in the membrane, right? I have potassium inside, I have sodium outside, right? What I'm going to do now is I'm going to take sodium in and potassium out. Look at this. I will take 3 sodium in exchange of two potassiums out. Exchange 3 for 2.
Just count.
It's counter, yes. So every time I move 3 sodium in, I'm moving 2 potassium out. I keep on doing that until we get what we need. You see this? I always do that exchange. Why There is a reason for that. We don't want to go deeper than that. It's the shape. The shape, the part of this, that's just if you wanted to know. But you have to remember the numbers. How did it work? Because this carrier, the one end that can fit sodium have three spots. And the other end that fits potassium has two spots only. So when you do this movement, 3 to 2. Okay? Another thing is called secondary active transport. So it is active transport, and you're using energy. But when you are using energy, you can take something on your way. Like this. If I drive my car, I put gas, I use my car, I'm going from Dearborn to Flint, all right? So is that active? Yes, I'm using my car, I'm using my gas, right? It's active. What if you tell me, like, can you take me on your way? Did you use anything? Did you use your gas? Did you use anything? No. I am the one who used it. So we call that secondary active. Secondary to active. So there is one that's active and you take me on your way. Like sodium, potassium like this. When it's doing this, glucose will say, sodium, are you moving in anyway? Yes. Carrier, are you moving sodium and you're using ATP anyway? Yes. How about if I jump and you take me with the sodium? Okay? So this is called secondary active. Secondary active. The third type is vesicular transport, and vesicular transport is coming from vesicle. And this is basically if this is something that's too big, like a truck moving in this classroom, you cannot. There's no way to move it in, right? You have to break down the wall. This is exactly the idea of vesicular transport. If there is something that's too big, you have to push it into the membrane. It's going to bend the membrane, detach, and move it in. I will show you guys the pictures. But you have to make a vesicle in order to do that. Clear? Break that down, break the wall, move the truck in, or break the wall, move the truck out. Clear? In both cases, you have to break down the wall. If you're breaking down the membrane of the cell, you're making a vesicle. I will show you the picture. But there are two different terms for this. Vesicular transport, vesicular transport, you're a vesicle transporting you across the cells. We call that cytosis. There is endocytosis and exocytosis. What's endo and exo? Endo means in, and exo out. So endocytosis, if you make a vesicle and move something in. Exocytosis, if you make a vesicle and move something out. Clear? Endocytosis, exocytosis. This is depending on, we're talking about the direction now. Are you moving in or moving out? But there is another thing. What are you moving? What's the nature of this big thing that I'm talking about? If this something is solid, we call it phagocytosis. Phago means eating. You eat food and you drink fluids, right? You eat the what do you drink food? No, right? So you have to give it the correct name. Panocytosis, pano means drink. Drink means it's fluid. It's a drink, fluid. So if the nature, if that's something that you want it to move in or out, if it is fluid, we call it panocytosis, which means drinking, as if the cell is drinking something in. If it is something solid, we call it phagocytosis. And there is a third one that's called receptor mediated. We will talk about it. But according to the nature, again, solid, phago means eat, fluid or liquid, peno means drink. The third one is receptor mediated. It's moving something in, but you have to interact with the receptor to allow you to go in, unlike the other ones. So look at this. You want it to move something in. Can you move in? No, you cannot. It's too big. Okay, how about I'm going to make this. I'm going to make a vesicle. The vesicle detach. moving what we need in, and then reform it. So here it is again, peno and phagocytosis. Drink and eat. Fluid and solids. Exocytosis is the opposite, moving something out. So here is what we talked about already, just reviewing. I'm not going to go through it again. We don't have time for that. You guys can take a look at it if you want to, but I told you what you have to know. Okay. Now, the membrane of the cells, they found out that, remember here I told you there is potassium and sodium inside, and there is potassium and sodium outside. Is it equal amount? No, it's not equal. It's different. So here you have potassium, which is cation, potassium, and outside you have sodium. Okay. Do you also have potassium outside? Yes. Just less. Do I have sodium inside? Yes, but less. Okay. So sodium is more inside than outside. Clear? Compare this to this. And compare this to this. Sodium is more outside than inside. This is number one. Inside you have cations and anions. Do you remember those? Positively charged and negatively charged. Inside and outside. So what's the difference between those two? They found out that overall, if I added all the positives and negatives inside, and the outside I added all negatives and all positives together, it's not equal. They found that inside is negative if compared to the outside. And we call that the membrane potential. And they found it out like this. They got a cell like this, and they get the voltmeter. Do you know the voltmeter? To measure the volt. So they put like one electrode inside like this, and the other one outside like this on the membrane. And they found there is a reading here. It's not zero. Which one is more negative? Inside is more negative than outside. Clear? Does it mean that I only have negative inside? I didn't say that. Okay? There is positives and negatives inside. There are positives and negatives outside, but it is not equal. More inside is negative as compared to the outside, and we call that membrane potential, and that's why you always see it negative like this. It has to be negative. Inside compared to the outside. Hmm? No, we don't say it this way. We just compare. If you look at the inside only, you have positives and negatives. That's it. How about the outside? Same thing. It's only by comparison. Comparing inside to the outside, it's negative. So we say negative 10, negative 70, whatever it is. Next thing is the cell life cycle. And you guys did that in biology, right? So I will go through it still, but I'll go faster. So cell division, the cells can divide in two different ways. Are you talking about a regular body cell, somatic cell, or are you talking about reproductive cells? Do you remember those? There are two different ways for reproduction. But there is one thing that's, do you guys remember the autolysis? What's autolysis? That was yesterday. What's autolysis? By itself. Yes, yes. So autolysis means if this cell is not working anymore, if you don't need it anymore, you don't just leave it. No, you have to break it down. Lysosomes open, release the enzymes, break down the cells. Remember this? This happens automatically in different types of cells, and it is called programmed cell death. Programmed cell death. The name for that is apoptosis. You need to remember this term, apoptosis. Apoptosis is programmed cell death. Is that something good or bad? It's actually good. It is good. Yeah, the cells are dying. Yes, good. Why? Because at the end of the life, let's say red blood cells. Red blood cells lifespan is 120 days. which is 4 months. All right. So red blood cells are working 4 months. After the 4 months, it's aged. It doesn't do the work anymore. There is no need to keep it. So guess what? It's going to destroy itself. Why don't you just leave it? Because first of all, it's not doing the work. Number two, if you leave it, it can start damaging other things and it can actually change to tumor or cancer if it goes beyond its lifespan. So this apoptosis is actually something that's protective. And guess what? If you lose it, you get cancer. One of the reasons for cancer is apoptosis didn't work. This cell is there and divide. What is the lifespan? It's supposed to die. No, it's not dying. There's no apoptosis. More tumor. So apoptosis is something actually good. Programmed cell death. Open the lysosomes, release, autolysis, apoptosis. So every cell is going to have its life cycle. Part of it is the interface. Do you guys still remember that from biology? You did biology, right? 131. Okay, so interface is that phase in between the divisions, which is the major part. Okay, so the cells are living, just working. At certain time, it's going to divide and then go back to the normal life, right? So interface is that majority of the life of the cells, of the somatic cells specifically, where the cells are not dividing. And it is called G0, G1, S, and G2. This is the subdivisions. Okay, so 0 means you're not doing anything. G1, this is where you're going to start, uh, the cells are going to grow, but it will start to duplicate organelles and start to do protein synthesis. Okay, G0, you're not doing anything. G1, do you need to know that specifically? I wouldn't say it if you don't need to know it. Okay, G0, you didn't do anything yet. G1, I'm going to duplicate my organelles and I'm going to make more protein. So are you starting to prepare? Yes, you're starting to prepare for division. And then you go to the S phase. What's S phase? S is the first letter of synthesis, protein synthesis. This is where you make protein synthesis. G2, continue protein synthesis until you get all what you need, plus you finished replicating the DNA. Do you know what replication means? Replication is what? Copying what? What to what? Uh, no, copying the DNA. Making what? This is transcription. Duplicating DNA to DNA. Just making more DNA. I have 23 DNA. I make it 46. Right? This is called replication. Making messenger RNA is transcription. Don't forget this. Okay, so G2, you're finishing the DNA replication, you're finishing the protein synthesis, you make the centrioles, and now everything is ready for you to divide after G2. Okay? I doubled my protein, I doubled my DNA, I doubled the organelles, right? Everything is double now. So now I'm ready for cell division, the actual division, and this is called mitosis. Mitosis. What do you call the cell division in reproductive cells? Meiosis. Yes. So this is mitosis, and mitosis start from prophase, metaphase, anaphase, and then telophase. Okay. There is part of it that's called cytokinesis after that. So let's start, you are in the interface and you go G0, 1, S, G2. Now you're ready. What are you going to do? I'm going to do DNA replication. How to do the replication? Is that different from transcription? Yes. If you make a copy in the form of messenger RNA, this is transcription. And what's your enzyme? RNA polymerase. Yes. Here the enzyme is DNA polymerase. Do you remember this? The product is the first part. RNA polymerase, you're making RNA. DNA polymerase, you're making DNA. So this is replication, you're duplicating your DNA. How do you do that? Same way as we mentioned in the messenger RNA, but here is a huge difference. Huge difference. Remember when we did transcription, the messenger RNA, you're just taking a copy of one protein only, right? But this, you're making an entire copy of the whole DNA, okay? So you're not going to do this piece only, you do the whole thing. So your DNA polymerase is going to make a copy of your DNA. You open it and you make a copy. And the other thing that's different here is, here are the two strands, right? Unwind, untwist. Now I have two, right? Remember in transcription, there is a coding strand, and the other one is called template, right? We make a copy of the template, a copy of a piece of it, not even the whole thing, right? In this one, the two strands, I'm going to make a copy of this and a copy of this. A copy of this and keep the copy with the half that's original. Like let's say this is a coding strand, okay? Coding strand. You make a copy and keep it with it. And then the template, you make a copy and keep it with it. Why are you doing that? Because I want to make sure that there is one original and one new one. Got it? And we call that semi-conservative. 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 Semi-conservative means, conservative means the two copies stay as it is. Okay? Okay, it goes like this. I have two papers, structureless. I have two structureless. Okay? It's a very nice copy. And I have two students. Both of them need it. And I want it to be fair. I'm not going to give one of them the two original copies that looks good, and then the other ones that looks like kind of sloppy or not perfect, I'm not going to give it to the other one. No, I give you one original and one original. You get one copy and you get one copy. So you got one original and one copy. It's semi-conservative because I give you half of the original. Did you get the idea? This is what it is. So everyone, we started with one DNA and we ended up having two DNA, but each one of them contain original and copy. We call it semi-conservative. Um, this helix, there is an enzyme that help you to separate them, and this is called helicase. Let me see if we have it or not. Uh, and ligase, yes, helicase and ligase. So what's the main enzyme that help us to do the replication? DNA polymerase. Clear? The one that help you to separate them is called helicase. And then this is original, and this is a copy. This is a double strand now, right? They should stay with each other, right? With hydrogen bond. But the pieces that you are making in order to fuse it together and make a whole strand, it's called ligase or ligase. Ligase is coming from what, if you forgot it? When you ligate something like this, ligated with this. Here it is. Ligation is to connect them. Helicase, unseparate them. Ligase, yes. Ligase, ligation. Are we familiar with this word? Ligate something? Like you connect them. Ligation. So here is mitosis, and mitosis will come from, we start from, um, first part is prophase. I know that you guys know this, but I will go through it fast. So prophase, pro means beginning, the very start. Prophase, there is early prophase and late prophase. In early prophase, you have two pairs of centrioles, and you start making the spindle fibers. Okay, centriole here, Every one of them will send fibers so that the chromosomes are going to be separated later on. This is early, late, so early and then late. Um, so the chromosomes are going to attach to the spindles. Each one spindle attached to chromatid. What's a chromatid? Have the chromosome. It's like this. This is a chromosome. This is chromatid, and this is chromatid. Together is like this. This is how the chromosomes looks like. Okay? Chromatid and chromatid. Two sister chromatids. Okay? So late prophase is where you're linking the threads to the individual or to the sister chromatids. Metaphase. Simply, meta means middle. You arrange them across the middle line. It actually means equator. Equator. Do we know what's the equator? The, exactly in the middle of the earth. If you put like exactly in the middle, we call that the equator. Middle line, whatever you want to call it. They arrange the chromosomes along the middle line. Anaphase, ana, Greek, for separation. Ana means separation. So anaphase is when you separate the two daughter chromosomes. You separate them from each other. Telo is a Greek for distance. Do you know what telephone means? Telephone? Telo means distance. Phone means sound. You know why they call it a telephone? For this reason. When you use your telephone, this is where the name came from. It's sound in distance. You are here, I'm in another state, we're talking. The sound is transmitted in a distance. Telephone. So telophase, meaning there is a distance here. And there is a cleavage furrow that form in between those two. And then cytokinesis is cyto means cells, kinesis means movement. Kinetic, it's coming from kinetic. So cytokinesis means the new cells, move away from each other. So now we have the two cells, and this is the whole entire process. The rate of mitosis, how many times do we need to know this mitosis? And mitosis is duplication or replication of which cells? Somatic or somatic cells, right? Somatic cells. How fast do we need to do it? Do we do it all the time? We do it sometimes. It all depends on which cells are you talking about. Like your skin cells, every day. Stomach, every day. Okay? Muscles, occasionally. Sometimes the muscles will never replicate. So it all depends on the need. If something like skin friction die every day, so you have to replace it. This is fast mitosis. So it all depends on uh, uh, your use and your needs. And there are factors that you use to speed it up or slow it down. These factors, one of them is called M phase promoting factor, and there are other growth factors. Those two are going to speed it up. So I have more in those in the skin and in my stomach. I have more of those, less in other things. And there is one which is doing the opposite, which is repressor gene and telomeres. When they are worn out, this will slow it down. So there are things to speed it up and others to slow it down. If it goes out of control and increasing like replication beyond your need, bottom line, beyond your need. Do we replicate? Yes. You just replicate according to the need. I lost 100 cells, I make 100 cells. That's it, right? I replace what I lost. That's it. So if you replicate within your need, that's normal. If you replicate beyond your need, this is called tumor. And tumor can be benign or can be malignant. I'm not going to go deeper. I just want you to understand this. The most important thing. Benign tumor is confined. It's covered. And this cover make it grow. It's okay to grow, but it does not go beyond its spot. So this tumor, let's say here is a tumor of the breast, for example. Now this is a benign tumor. The benign tumor is a tumor that form like this, okay? And then it's become bigger. It's surrounded by a membrane. It doesn't go anywhere. Stay in the spot. So it grow bigger, 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 bigger. Just the size. Is it ever going to exceed this? No, it does not. It will not. It will not. If it rupture, it's not going to spread. But usually, it just expands on the expense of the surrounding tissues. Okay. What's the key line here is confined. It doesn't go beyond that because it's surrounded by a membrane. Malignant, the key word is invasion. You go beyond that. What if the tumor is getting bigger? Still benign, it's okay. If it's getting big, is it invading or not? That's it. So let's say in the case of breast cancer, did it leave the breast and started to go to the chest, like go to the muscles? If not, it's benign. Okay? So this is the bottom line. There are other things, but this is the most important one. So here is a cancer, and there are genes that stop that from happening. If these genes are affected by something, it will start malignancy. Something like what? Any irritant. Like you know why they put like this? If you when you do X-ray at the dentist, they put this shield or they put this cover because if you do it several times, this radiation can go actually to the genes and change their nature. So the oncogenes, onco is a prefix for tumor. The oncogenes are the genes that stop the tumor. If you break it down, it will form a tumor. There are other things that are called carcinogens, mutagens. Carcino is cancer. Gen means generator. Things that can generate cancer. Example, hepatitis C. If somebody's having hepatitis C in the liver, and if it is chronic, it can actually end up having cancer. Um, HPV, that's an excellent example. Cancer cervix, do we know that? And that's why ladies should be tested for that virus because it's an oncogene. Okay, I mean it's a carcinogen. Metastasis means you don't only invade, you reach the blood or you reach fluid and go to a distant organ. Cancer lung, and then this person is getting cancer in a bone somewhere else. Okay, so this is called metastasis. Look at the metastasis. If it reaches the blood, it's going to go somewhere else. So if the cells, we call the cells differentiated and undifferentiated. Differentiated means if the cells are growing within the normal pace, like one cell makes two, make four, eight, according to your needs only, that's okay. You have the time to make perfect cells. The perfect cells are called differentiated cells. Okay? Good cells. But if you go faster, the cells become undifferentiated. Translation. I give you practical example. If I ask you, I give you everything from scratch, flour and eggs and everything, and I ask you, make 5 pizza for me. You can make 5. You have the time to do it, right? 10, you can do it. 20, maybe. If I ask you to do 500 pizza, you don't have the time, right? You don't have the time to do all of this. You just get some flour and just hear this, right? It's not ready yet. It's not pizza yet. So when you say a pizza that's ready to eat, this is differentiated cells. If I push you to go faster, like in cancer, faster, 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 you get cells that are not differentiated. You look at it, what is this? This is supposed to be like lung cells. It doesn't look like lung cells. Yes, you didn't give it the time to differentiate. Okay, it's not pizza yet. Okay, so